Hello everyone, Doc Ron Bio here to discuss basic genetics with you. We will start at the beginning. In the beginning, as far as genetics goes, was Gregor Mendel. Before Mendel, humans knew you could manipulate organisms for desirable traits. The artificial selection of crops for desirable traits was the basis of agriculture. Mendel put these ideas to the test experimentally, and as a result, we called these findings the law of Mendelian inheritance. These laws explain how traits are transmitted from one generation to the next in a predictable way. Mendel is an interesting figure, actually. We don't have time to go into the details of his life in this video. He was an Austrian monk who wanted to figure out how traits are passed from one generation to the next. He used pea plants in this process in a garden in the abbey that he was at. So unfortunately for Mendel, the significance of his genetics experiments were not recognized for decades. Luckily for us, they eventually were, and they helped to guide the modern genetics understandings that we use today. So let's take a look at some basic pea plant crosses and establish some vocabulary. We don't have the time to go into detail on how one performs a genetic cross in pea plants. Let's just say that it's challenging because the pea plants can self-fertilize. True breeding plants are plants that, when crossed to themselves, result in the same phenotypic traits. So for example, if you were to cross these violet flower petal pea plants to violet flowers with violet petals, they would produce offspring with violet petals. Same thing for this white flower plant. If you were to cross it to another plant that had white flowers, they would produce offspring with white flowers. So that's the basic definition of true breeding. Recall that phenotypes mean the physical appearances that we're talking about. So when a purple plant is crossed to a purple plant, that phenotype for flower petals is the color purple. Okay. Interestingly, when you take a flower petal plant, a purple flower petal plant, and cross it to a white flower petal plant, in what we call the P generation here, that is the parental cross, all of the offspring in the next generation, that is to say the F1 or filial 1 generation, are all phenotypically purple. So the fact that all the offspring of the P cross are purple shows that the genetic material that creates purple phenotypes for flowers is dominant to the genetic material that produces white flowers. Okay, so now there's this idea of dominance emerging in a cross. A quick note, we know that the genetic material in a gene is a gene on a chromosome, and that the two different versions of a gene are called alleles. But recall that Mendel's experiments predated knowledge of the gene, so that's why I describe it in that manner. From here on out, though, I will describe things as genes and alleles. So dominance is a really interesting word here. Most students imagine a battle or show of force on the part of the allele that causes purple, when in actuality what this really means is that the dominant allele masks or covers the recessive allele phenotype. In this case, you can't see white flowers when purple coloration is produced. So we say that the purple color, and more importantly, the allele that creates that purple color, is dominant. Okay. We note dominant alleles with a capital letter. So here, if we were noting purple, the capital P here would note purple. And for recessive letters, like this lowercase p here that has a little underline on it, so it's not confused with the capital letter, the lowercase letters are used for recessive alleles. When it comes to Mendelian genetics, the two alleles noted are one or the other. This is partially what makes Mendelian genetics so predictable. Mendel then let the F1 offspring self-pollinate. So here we're seeing these F1 purple flower plants. He let them self-pollinate, self-fertilize. So it's a purple plant crossed to a purple plant. Okay. When these purple hybrids create offspring, you see the return of the parental white in the offspring. Okay, another quick note here. The reason we call this a monohybrid cross is that here in the F1, we let the plants self-fertilize. So Mendel called these plants hybrids. We refer to these plants as heterozygotes. So we're going to let, in this generation, these plants self-fertilize. Okay, the cross is for two plants that are heterozygous for the one gene being considered. So it has a capital P and a lowercase p. I will explain how this works in this slide, which has a little more detail. Note that you're switching from the flower petal color gene and associated phenotypes to the pea seed color gene and its associated phenotypes. The genetics works the same, we're just switching from a different trait. So we are still only considering one gene on one chromosome in this series of crosses. This gene, like in the previous example, has two alleles. One is dominant, we call that one capital Y, and the other is recessive, and that's lowercase y. Okay, so this slide shows you the results of meiosis and how the possible alleles are packaged into gametes. So remember, genetics really factors in meiosis and how alleles for genes are packaged into gametes. Okay, so here's an example. If your genotype or genetic makeup as a diploid organism is capital Y, meaning your two homologous chromosomes have a dominant allele for the pea seed color gene, okay, so it's homozygous dominant. If this is the case, 100% of the gametes from this organism will have the dominant capital Y allele for the pea seed color gene in gametes. Okay. Likewise, for the plant producing green peas, which has a homozygous recessive genotype, this plant will only package recessive lowercase y's into the gametes. So this is a very straightforward, because one plant only contributes capital Y and the other only contributes lowercase y's. 100% of the offspring will be heterozygous or contain one of each alleles. Okay, so this plant only contributes capital Y, this plant only contributes lowercase y, and when these gametes come together, you have a heterozygous plant that has one capital and one lowercase y allele. Interestingly, all F1 plants make yellow seeds. So again, we're calling in the idea of dominance here. The allele that leads to yellow peas, the capital Y allele here, is dominant to the lowercase y allele. Okay. Again, if we're letting the offspring created by this parental cross self-fertilize, we see a heterozygous individual cross to a heterozygous individual, and what's not shown there is the other offspring. So it's, capital Y, lowercase y, heterozygote cross to capital Y, lowercase y, heterozygous individual. Again, this is where we get the idea of the monohybrid cross, because you're crossing two heterozygotes together. For each of these heterozygotes, there's a 50% chance of the gamete obtaining a chromosome with a capital Y allele for pea seed color that is yellow, and a 50% chance of the gamete obtaining a chromosome with the lowercase y allele, which leads to greenness. And we show that here, okay? So 50% chance of this, 50% chance of this in both offspring, both plants that we're crossing. Okay. So lastly, this monohybrid cross results in predictable genotypes and phenotypes in the offspring. So if we were to express the results as a ratio, one would easily be able to predict what we saw in the offspring. So you can see the results of this cross here. So when it comes to genotypes, it is a predictable 1 to 2 to 1 ratio. That is, one homozygous dominant, two heterozygotes, and 1 homozygous recessive. So for the genotypic ratio, it's a predictable 1 to 2 to 1 in monohybrid crosses. Likewise, with the phenotypic ratio, it is also predictable. So here you can see the phenotypes, and here it's a predictable 3 to 1. That is to say, all of these that contain at least one dominant allele will be yellow, and the homozygous recessive individual that does not have any dominant alleles, it will be the only lone green one. So you'll see that in a 3 to 1 ratio. So if the population is very large, say it's 400 individuals, you would expect to see 300 plants that are yellow, 100 that are green, a 3 to 1 ratio. That's about it for now. I think one of the great things about the monohybrid cross is it allows you to practice vocabulary. So here you have to remember, know the difference between genotype and phenotype. We really emphasize that in this video. So know genotype, genetic makeup, phenotype, physical appearance. And then of those types of genotypes, know the difference between homozygous and heterozygous, okay? Keep this vocabulary in mind as we work on future examples, and I'll see you next time.